Yeah, so I want to give a presentation on some of the work that we've done on artificial nucleic acid backbones. And I want to talk about synthetic biology, but also a little bit about therapeutics, where the therapeutic area is going, if I get time to do that. So we'll see how we get. So first of all, I think it's important to say that the work that we do is based on oligonucleotide synthesis, and we have a lot of many years of experience in making modified oligonucleotides, and they're all made basically by the phosphoramidite method. And I'm not going to talk about chemical synthesis in the talk. Excuse me, just 1 minute. This is, yeah, it's okay. I'm not going to talk too much about, I'm not going to talk about chemical synthesis, but I do want to mention that this phosphoramidite method, which was developed by Marv Carruthers in the 1980s, it kind of underpins everything that we do, and not just us, but almost everybody who works in biology now. And it's important to recognize that. So I wanted to give a bit of a, acknowledgement to that. A lot of the work that we have done was funded by BBSRC on an SLOLA grant, which is now expired. This is Marv Carruthers, and he I think will be, I'm sure he'll be a prime candidate for the Chemistry Nobel Prize, the next Chemistry Nobel Prize, because of the importance of this area to biology in general, but also to things like vaccines and messenger RNA technologies. And that's going to be a big focus going forward in terms of recognition. But what I want to talk about myself in terms of our work is looking at chemical ligation, how we can make artificial nucleic acid backbones, and what the properties of these artificial backbones are. If we want to join together nucleic acid strands, then in nature, this is done by ligase enzymes. And they repair broken pieces of DNA. And they're involved in recombination and replication, but they're not generally adopted by people who synthesize genes. There are good reasons for that. PCR dominates this area, but ligases are important enzymes. But they do function under a limited range of conditions. You can't really use ligase enzymes on artificial nucleic acids, with a few exceptions. And the kind of conditions are very limited. The enzymes need very specific conditions. So they're not always compatible with modified nucleic acids and other chemistry. So we thought we would try and work out methods of joining DNA strands together chemically that will be compatible with artificial nucleic acids systems. Now, early work was done in this area in the late 1980s and 1990s, where people developed a method based on ligating together a 3' hydroxyl and a 5' hydroxyl to form the phosphate bond between oligonucleotides, using splints to join the oligonucleotides together. So this work didn't go very far because that chemical reaction is quite a difficult one actually to do, and it's not very high yielding. But it does give you natural nucleic acids, which is, in one hand, is great, but on the other hand, chemistry is not efficient. So we wanted to go in a very different direction. We wanted to develop artificial nucleic acid backbones. And the reaction that we decided to pursue was a reaction which was developed in the early 2000s, which is a click reaction between an alkyne and an azide to form a completely artificial backbone, in this case a triazole. So the idea is that you take an oligonucleotide with an alkyne, an oligonucleotide with an azide, add a catalyst to start the reaction to form this artificial backbone. The reason that we use this methodology, or started this methodology, is because azides and alkynes are easy to attach to oligonucleotides chemically, and they're very selective in their reactivity with each other in the presence of compound. The reaction itself works well in water, in aqueous buffer, particularly well in aqueous buffer, which most organic chemical reactions don't. They're often spoiled by water. And again, you can switch the reaction on whenever you want just by adding copper ions. And the resultant triazole is chemically very stable, which is useful. Now, this chemistry, that we're going to use was developed, as I said, in the early 2000s, and it was the Nobel Prize this year went to click chemistry, and Barry Sharpless and Morton Meldal developed this CuAAC reaction between an alkyne and an azide, which has had great benefit across a wide range of different areas in materials, chemistry, biology. If we join together two nucleic acids and we create this kind of backbone, we have to think about its properties. Well, first of all, there are a little bit of similarities with normal phosphodiesters, but largely it's very different. So it doesn't look on the face of it if it's going to be a very good analogue of a phosphate bond. Now, this wasn't the only artificial nucleic acid backbone that we made, but a bit like evolution, it's the one that survived the last 20 years, and I'll explain why that is. First thing to do was to see if we can actually if we can assemble DNA strands using this kind of chemistry. So what we did for this was to take 300 oligonucleotides, which have got alkynes and azides at specific positions, and template them with complementary oligonucleotides to hold them together in the right order. And then add copper 1 and see if we can get the product. And then see if we can amplify the product by PCR. So the idea here is we link together these three strands. Well, first of all, joining them together was very efficient. And when we added PCR primers, we were able to demonstrate that we've got a very clean PCR product. So what this is telling us is that when we assemble oligonucleotides to form bigger species, and these species contain triazoles, the DNA polymerase will quite happily pass through the triazoles and copy the DNA strand. It's okay to show a gel, which shows bands on a gel, but what's really important is to sequence the DNA, to see if the DNA itself has got the right sequence, the complementary sequence to the template. And we did many examples of this. I'm just showing you some raw data here, but we were able to show that when we link together C with C, or C with T, or T with T, whenever we linked together these strands and carried out Sanger sequencing, we always got the correct product. The sequence around the triazole was the correct sequence. There was no skipping or misreading or anything like that. And we did over 100 clones and sequenced over 100 clones to prove this. We had to do that for publication. But it was quite a surprise in some respects, but we were able to show that the triazole didn't inhibit the polymerase from reading the correct sequence. We also did rolling circle, well, just before I do that, this is just to show that okay, so first of all, yeah, we did rolling circle amplification. We took 100 DNA, which is shown here, which has got an alkyne at one end and an azide at the other end. We added copper ions and it cyclized to form a cyclic 100 with the right mass. And we did rolling circle amplification around that single-stranded template to produce very large quantities of product from this cyclic template that contained a triazole. The point here is that the enzyme has got to read through the triazole many, many times to make a very long piece of DNA. And we were able to make DNA nanoflowers from this kind of cyclic construct. And this is just a gel to show that the amount of DNA that we got from the triazole template was very similar to what we got from a phosphodiester template. So this is continuous read through of the triazole to produce a very big piece of DNA. Now, PCR is obviously useful, a very incredibly useful technique, but to use it as an analytical technique to see if you can read through an artificial DNA backbone is actually a little bit kind of questionable, because if you make a bit of template, the reaction will take off, and then you'll be reading through always an unmodified template. So it's not necessarily a critical test of how well the triazole can be read through. A very good test is to take a single-stranded DNA template and just read through it once by linear copying with one primer, and to do that as a function of time and see how much product that you get. And when we did that with a triazole DNA template and a normal template, they both gave, within 3 minutes, a very large amount of the correct amount. They consumed all the primer to produce the product. So we now know that the process is happening in a reasonable timescale to a reasonable amount of efficiency. I have to say that if we take this template and we read through it and look at it after, say, 10 seconds, there's a big difference between the unmodified template and the triazole template. The triazole is causing the polymerase to pause, but eventually read through and get to the end. Just to mention also that when we transcribe a piece of DNA that contains a triazole linkage in the coding region, then if we compare the triazole template with the unmodified DNA template, the triazole template produces almost as much RNA as the unmodified template. And this shows that RNA polymerase can also read through this triazole template. So we've got replication and transcription. And this is just mass spec proof that the transcript from the normal template control and the triazole template control give the same product. It's the correct product. It's got the correct length, the correct mass. But we wanted to also see if this could work within a living system. So we took a E. coli and we put a plasmid in E. coli. We put the click linkage within the plasmid in E. coli. This time we put one in each strand. So we have a plasmid that contains 2 triazoles, and we express that plasmid in bacteria. And the plasmid contains an essential gene for bacteria. So the bacteria can't grow if the plasmid is not expressed. And we looked at the result of that experiment. And what we found was that, again, if you compare the triazole DNA plasmid with the normal plasmid, we got essentially the same amount of colonies from both. And we sequenced the DNA that came out of it, and it was correct sequence. So that just tells us that if you put these triazoles into E. coli, the polymerases within that system can also read through the triazoles. If we took a strain of E. coli that's deficient in DNA repair, we still got a really good result, 92% of the number of colonies, which tells us that this is not some kind of weird DNA repair kind of thing that's going on. The triazole is almost certainly being treated as if it was a phosphodiester within the bacteria, by the bacterial polymerases. We did the same thing, just briefly to mention, we did the same thing with a non-essential gene in the bacteria, which is an M-cherry gene. So we put M-cherry into plasmid into bacteria, and we got fluorescent colonies in a similar amount to the fluorescent colonies from the positive control that doesn't contain a triazole. So whether it's an essential gene or a non-essential gene, the bacteria were happy to utilize the DNA that contains the triazole and make RNA from it. So that's all good. And the next step was to put a plasmid into a human cell. So we used NCF7 cells, and we put M-cherry in there, and the plasmid was able to be transcribed to make fluorescent cells, effectively, to express the M-cherry in these eukaryotic cells. So eukaryotic cells also behave correctly with this kind of triazole DNA. So we did some biophysical studies in order to work out what effect the triazole has on the structure of DNA. To do this, we took a short oligonucleotide duplex that contains a single triazole linkage between 2 thymines, as I've shown here. And we studied the structure of this duplex by NMR. And the NMR analysis told us that the duplex has generally the same shape as an unmodified duplex. Looking at NOEs, looking at the protons around the triazole site, we were able to show that there is a displacement of the DNA backbone around the triazole. The triazole link is a bit too long compared to a phosphodiester linkage. And there is some fraying of the base pair around the triazole. So the NMR study told us that there is a little bit of a displacement around the triazole in terms of structure, and that we do get instability around the triazole linkage. So we produced a model of that, which you can see here. The triazole you can see sticking out here, but the general DNA duplex has got generally the right shape with a little bit of distortion around. Let me show you that one again. You can see the triazole in red compared to the other strand, which is the control strand, which doesn't contain a triazole. So this is an overlay of a triazole DNA against the normal DNA, and it's an NMR structure. So it tells us there's a little bit of distortion and a little bit of instability. But one thing that's interesting, which I think is on the next emphasize, is that one of the Let's get this right, it's just stalled a little bit. There we are. One of the phosphate oxygens of the phosphodiester of normal DNA is quite close to one of the nitrogens of the triazole when you overlay it with the triazole containing DNA. And these are both potential hydrogen bond acceptors. So it means that it's possible that the triazole nitrogen, okay, it's all right. The triazole nitrogen is acting as a hydrogen bond acceptor, because when DNA binds to DNA polymerase to read through the DNA template, hydrogen bonding is quite important. So this is looking at approximately the number of nucleotides in a DNA template that would be bound at any time to a DNA polymerase. And when the polymerase binds to the DNA template, amino acids in the protein form hydrogen bonding interactions with the DNA. There are some electrostatic interactions, but they're mostly hydrogen bonding. And if the triazole, first of all, the triazole is only 1 out of 10 nucleotides bound to the polymerase, so it's only a tenth anyway, approximately, and it can actually form, in principle, hydrogen bonds. So the polymerase doesn't seem to be that selective about what's happening at the backbone. We took this idea of joining DNA strands together using triazoles a little bit further. We took a whole series of oligonucleotides, which had got alkynes and azides in them at the ends, and we templated them with complementary DNA, and we carried out a click reaction to make a very small gene, ILOV, which is in the 300s, an ILOV gene, which contains 8 triazoles. And we were able to show that when we assembled that gene, that it can be expressed in E. coli. So we assembled a full gene, albeit a very small gene, using this methodology, and used it to make a protein in E. coli. We've since gone on to make bigger genes than this, but this was the first example that we did, which we published in 2017. Now, I mentioned that the triazole linkage can form hydrogen bonds with proteins, but if you actually methylate the triazole here, then the hydrogen bonding capacity is strongly inhibited. It's also a positive charge now in the backbone, as opposed to a negative charge, which you get in normal DNA. So we did that. We synthesized DNA strand which had got a methylated triazole. We did it by making a phosphoramidite monomer and making the DNA strand with a methylated triazole. And we studied the properties of the methylated triazole compared to the unmethylated triazole. So we want to see what effect it has of blocking that nitrogen atom on the polymerase. And up until now, everything had worked really well. But at this point, we were actually quite happy that simple methylation of this nitrogen in the backbone really messed up the DNA polymerase. So these are 3 different thermostable DNA polymerases, and this is the sequencing results around the triazole. And what we're showing here is that in this case with GOTAC, we got some deletion of T and C and these 2 T's. With CodXL, we got also deletions in this area, and with Fusion, we got even stronger deletions. So just a simple addition of a single methyl group, it messed up the ability of the polymerase to treat that backbone as a normal backbone. We also wanted to work out what effect the triazole had on read-through in terms of the rate at which the polymerase can read through the triazole linkage. So we did a very simple experiment. We made triazole template, compared it with normal template, and we measured the CT value, or the CQ value, if you like, of the PCR reaction. The number of cycles it takes to make a significantly observable PCR product. And what we found was, well, the theory is this: if the triazole is read through quickly, there should be no delay in making the product. But if it's read, and that would give you a clean read-through. But if the triazole is slowing down the polymerase or causing it to pause, we should be able to measure that pause. by looking at the CT value in PCR. And what we found was we took a whole series of different triazole containing DNA templates, compared them to the normal phosphodiester, and looked at this CT value. And we looked at it in different sequence contexts. I'm showing TT and CT or GT here. And the effects were quite consistent. And in general terms, this is color-coded. So the normal phosphodiester is in grey, and these different triazoles are in different orange, yellow, and red colours. And what we found was that as we increased the length of the DNA backbone between the two sugars, the polymerase was slowed down more and more. So we got pausing of the DNA polymerase according to the length of the backbone. The backbone that I've been talking about most so far here is the orange one, which the CT value is very sensitive to the cycle time. You have to do the PCR more slowly if you want to get it to be really efficient. So the polymerase doesn't fully recognize the triazole as normal. It's slowed down by that modification. One thing about a triazole and click chemistry is that you can actually you can create a 1,4 triazole, which is the one I've been talking about so far, by using copper catalyzed click chemistry. But if you use ruthenium instead to catalyze the reaction, you make a 1,5 disubstituted triazole, which has got very different physical properties. The problem is that if you don't use the copper catalyzed reaction, you can't do it in water. So the copper catalyzed reaction is really great because you can use it under biological type conditions. But the other types of catalysis, they don't work under aqueous conditions. So in order to just test the theory about how well they read through, we had to incorporate these 1,5 disubstituted triazoles by adding as dimers into synthetic oligonucleotides. So we wanted to test how they responded, how the polymerases responded to these different triazoles, but we had to do it by using chemistry to assemble the DNA rather than ligation. When we did, this is quite a complicated slide, but I only want to make one specific point. We made a whole range of different triazoles again, like we did before, but the only one I really want to focus on is this one here. This one is a 1,5 triazole, and what we found was that linear copying of that triazole is extremely efficient. This is this one here. Very efficient. Many of the other triazoles, if you look at the actual copying of the triazole after certain time points, it's not finished. But this one, the one in yellow, which is the 1,5 triazole, it reads through very quickly. So this was published relatively recently. So now we have a triazole which works more efficiently than the one that we spent most of our time working on previously, but which is easy to make. It's the usual story. The one that's easy to make is not the best. And it's the same story in PCR. In PCR, if you take this 1,5 triazole here, it reads through, even at very short cycling times, it reads through really well. So for whatever reason, a polymerase is much happier with this triazole than some of the others. Now, a 1,5 triazole is more flexible than a 1,4 triazole because it only contains one bond going through the triazole. So the number of sp2 linkages is only one. So it's a much more flexible triazole linkage. And the length of it, which is 6 atoms, is closer to the length of a normal phosphate. It's just 1 atom longer, but the bonds are a bit shorter anyway, so it's a similar length to a normal phosphodiester, and it's quite flexible. So that was an interesting result that we got, changing the nature of the triazole. But we did another experiment at the same time, where we made an amide linkage in between two sugars. Now this linkage is quite different. It's a carbon-carbon, nitrogen-carbon linkage. So this linkage doesn't have any oxygens in the linkage between the two sugars, but it has almost exactly the same length as a phosphodiester, and it has a carbonyl group, like a phosphate, and an NH, which is also a hydrogen bonding residue, although it can be an acceptor or a donor. And this linkage is read through extremely well by DNA polymerases, just like that triazole that I showed you. But the problem with this linkage is it's extremely difficult to make chemically. So this is not a realistic linkage for a kind of a biotechnology application, because you have to form it under non-aqueous conditions pretty much, and the monomers, the building blocks, are quite lengthy to make. So although it can be used, it's quite demanding to work with. But that brings me to the last part of the talk, the second part, which I wanted to go into in terms of using oligonucleotides in therapeutics, because the amide backbone turned out to be really useful in a different context, and I'll explain that. First of all, oligonucleotides are becoming very, very important in therapeutics. There are now a large number of oligonucleotides in the clinic to treat different diseases. They basically bind to messenger RNA and either kill the messenger RNA or cause a change in mRNA assembly. There are 3 different therapeutic modes: antisense oligonucleotides in orange, which kill the messenger RNA; small interfering RNA in purple, which also destroy messenger RNA; and exon skipping oligonucleotides, which repair messenger RNA. So as you can see from this list of the 3 therapeutic modalities, there are a similar number of oligonucleotides in each that are now in the clinic. And they're used to treat genetic diseases, like, for example, Duchenne muscular dystrophy, spinal muscular atrophy, amyloidosis. But more recently, they've come into the clinic to treat relatively common diseases, like very high cholesterol. So this molecule inclisiran, which is an siRNA, can be used to treat the kind of high cholesterol that statins don't work with. And interestingly, you only have to inject the oligonucleotide into the individual once every 6 months to get the therapeutic effect, rather than taking a tablet every day. So this is a really exciting area now, therapeutic oligonucleotides. And one of the reasons it's so exciting is because small molecule drugs treat mostly proteins. But that only represents a tiny fraction of the RNA of the human genome. So only a tiny fraction of the RNA of the human genome makes proteins which can be treated by drugs. It's only about 1.5%. But the RNA in the human genome that's made by the human genome is so much more. It means that we can now target diseases either that don't make messenger RNA to make proteins, or whereby the messenger RNA isn't making a protein that can be drugged. So by attacking messenger RNA rather than proteins, the therapeutic potentials is increased enormously. And this is coming from a review in Nature Reviews in Drug Discovery. So mRNA, or RNA in general, is a really important target for drug molecules. And because DNA and RNA interactions are so predictable, then oligonucleotides are really attractive as potential drug molecules. Problem is that unmodified DNA isn't stable in cells. It's degraded by nuclease enzymes. It does have a good affinity for its target, e.g. messenger RNA, but toxicity can be an issue. But the really big problem is getting these relatively big molecules into cells. So getting oligonucleotides into cells and out of endosomes is an issue which needs to be tackled. So we wanted to look at our triazole backbone in this context. As I mentioned before, the problem is that triazoles cause instability when you bind triazole oligonucleotides to their RNA target. So here we have a melting curve showing that a normal DNA bound to RNA gives a certain duplex stability. When you add one triazole, the duplex stability falls. And that's not great for therapeutics because we want to bind messenger RNA and we don't want the oligonucleotide to fall off, basically. So this is an issue. Instability of the duplex is an issue. This is coming from our study on this short piece of DNA against RNA. But we know that we can increase duplex stability by introducing modified sugars, like, for example, locked nucleic acid, LNA. So if we put LNA into an oligonucleotide, it binds to its target much better than DNA because it enhances the A conformation, which makes DNA bind well to its target. So we looked at this. So we took an oligonucleotide which had got multiple triazoles, and it has a very low melting temperature compared to unmodified DNA. The melting temperature drops a lot. Adding a triazole at the 5' side didn't help. But adding a triazole at the 3' side did help. Sorry, an LNA at the 3' side did help. And adding 2 LNAs was highly beneficial. So we ended up with an oligonucleotide which had the same melting, which had a better duplex stability than the unmodified. So we've compensated for the instability of the triazole by adding locked sugars to the oligonucleotide to get a duplex which is stable, and we're continuing to work on this. We did the same thing with a carbamate linkage. So now we've got a linkage which has got 2 oxygens and a nitrogen, and we can either put the nitrogen on the 3' side or the 5' side, and the duplex becomes pretty unstable when we look at the stability of carbamate oligonucleotides, and it's much worse with this nitrogen here than when the nitrogen's here. So we can put a carbamate linkage in between 2 sugars, but it destabilizes the duplex. But when we introduce triazoles on both sides of the carbamate, we end up with an oligonucleotide that has a very good duplex stability compared to its DNA complement here. So this is the carbamate. This one here is the carbamate without LNA, and this is the carbamate with LNA. So again, an artificial backbone with a locked nucleic acid gives pretty good duplex stability. So the LNA has to be at least on the 3' side and preferably on both sides of the artificial backbone. Okay, so we've established now that we can put triazoles or carbamate linkages into oligonucleotides, and these linkages will form stable duplexes as long as we surround them with locked nucleic acid. So we wanted to also look at the amide. So the difference between the carbamate and the amide is just a single atom, right? So this is the carbamate with an oxygen, and this is the amide with the CH2. So the question is, if you just swap the oxygen for a CH2, how does that affect duplex stability? Well, this is a bit complicated, but this, all I'm showing on this slide is that what people generally do in the therapeutic oligonucleotide area is to make modified sugars and modified phosphorothioate backbones in order to make the oligonucleotide stable in cells, because normal DNA is not stable in cells. So very artificial backbones like PMO backbones and phosphorothioate backbones with modified sugars greatly stabilize the oligonucleotide against degradation, but they also make sure that the oligonucleotide binds well to its target RNA. So we wanted to look at these different backbones and compare them with an amide and a phosphorothioate. Now, the reason that we want phosphorothioates is very fundamental. When you look at oligonucleotides, which are used in therapeutics, they always have phosphorothioate backbones. And the phosphorothioate backbone enables the oligonucleotide to bind to serum proteins, and that complex can get into cells via endosomes. If you remove the phosphorothioate backbone, apart from making the oligonucleotide unstable to enzymatic degradation, it also doesn't go into cells properly. So what we wanted to do was to see if we reduced the negative charge of the oligonucleotide, get rid of some of the phosphothioates by introducing a neutral backbone, whether or not we could get improved cell uptake by balancing the negative charge, which is thought to be a bad thing in terms of cell uptake, but maintaining the sulfur, which is thought to be a good thing, and doing that by introducing amides within the oligonucleotide. So we want to reduce the negative charge, maintain sulfurs, and see if we get a different set of physical and biological properties. I'm not going to go into the chemistry of making these oligonucleotides, other than to say you can assemble them on solid phase from acids and amines, or you can incorporate them as phosphoramidite dimers. But this is a lot of work, but nevertheless, you can make these oligonucleotides in reasonable quantities. And this is an example of an oligonucleotide which contains 4 amides, LNA amides. And this is the HPLC and this is the mass spec, just to show that we can make them, purify them, and get them in reasonable amounts. So this experiment tells how we compared the stability of oligonucleotides containing LNA with amides and compared it to oligonucleotides which had either got no LNA, 1 LNA, or a normal phosphodiester. And what we found was that if we take oligonucleotides with different numbers of LNAs, that the oligonucleotide that contained LNA amide gave a very stable duplex. Just working out to get the color code correct, which is shown here. And as we increased the number of LNA amides, the oligonucleotide duplex became more stable. So unlike any previous backbone that we've studied, the LNA amide makes the DNA more stable. It just doesn't compensate for instability. It makes it more stable. So what the story here is that just by changing this, putting a carbon here instead of an oxygen, gives much more stable DNA duplexes. And the LNA strongly contributes to duplex stability. Now, there is a caveat. The caveat is that if we make an oligonucleotide which has just got LNA without amides, it's even more stable, right? But it's, in a sense, too stable. So we've reached a level of stability which is really useful by combining LNA and amide in the same dinucleotide unit. And again, we wanted to work out what kind of effect these different modifications have on the stability of a duplex, but also the structure of a duplex. So with James Hall, who's a collaborator at Reading University, we synthesized a whole series of DNA-RNA hybrids in which we incorporated the modification in one place here, which is shown here in red, and we compared the structures of different combinations where this dinucleotide step had a normal phosphate, where it had a DNA amide, where it had an LNA amide with the LNA just on top, or an LNA amide with the LNA in both positions. So this is the most extreme example, where we have LNA on both sides of the amide, and we compared it all the way back to the unmodified DNA-RNA hybrid. And the individual structures are shown here, with the position of modification highlighted. This is the unmodified. And what you see here is that they all look pretty similar. And when you overlay them, it just emphasizes there's almost no difference in the structure. These are X-ray structures at high resolution of these different DNA duplexes. And when you look down on the helix, there's not a lot of difference. So in other words, the oligonucleotide that we've synthesized with the LNA amides is able to adopt exactly the same shape within a pretty much, a little bit of error, as an unmodified DNA-RNA hybrid. So entropically, it's in the right kind of shape to bind to its target. And that must be one of the reasons why it forms such stable duplexes. So in a sense, the X-ray structures were all identical, but in a sense, that's also a good thing. So this will just, the last little bit of the talk now is what happens when you make an oligonucleotide and put it into cells that contain these modifications. So this is a study of luciferase activity in HeLa cells. Now, the way this assay works is that we have a luciferase gene in HeLa cells, and the luciferase gene is not functional because the splicing is incorrect. So what you do is you bind an oligonucleotide across a splice junction, and when it binds, it restores correct splicing to the luciferase gene and produces a light signal. So in the absence of an oligonucleotide, these cells don't express luciferase. But when you bind an oligonucleotide in a specific place on the pre-messenger RNA that has to make the messenger RNA, the splicing becomes correct and the cells express luciferase. So we took this assay and we carried it out with a scrambled oligonucleotide, an oligonucleotide that doesn't contain phosphorothioates, an oligonucleotide that doesn't, two oligonucleotides that don't contain phosphorothioates, an oligonucleotide that contains the amides and phosphorothioates, and an oligonucleotide that just contains LNA, so lots of controls, and an oligonucleotide that contains phosphorothioates with no LNA. So what we're saying is that we have an oligonucleotide that contains phosphorothioates, which is here, which works, an oligonucleotide that contains LNA and phosphorothioates, which also works, and an oligonucleotide that contains amides, LNA and phosphorothioate, which also works. But none of the negative controls work at all because they don't contain phosphorothioates or the scramble. These three all behave the same, effectively, within error. But the thing is that, and they're also, should I also say, they're also concentration dependent as well. So they all work, and they all work in a concentration dependent fashion. But this is by transfection. So this is taking these oligonucleotides, adding them to the medium with a transfecting agent, which is basically a lipofectamine transfecting agent. All the oligonucleotides get into the cell, all these three, the phosphorothioate ones, get into the cell, and they all work. Now, when we repeat this experiment, but now we don't use a transfection agent, we just rely on the oligonucleotide's ability to go into the cell, we have a very different result. And the result is the only oligonucleotide that really works is the one that contains the amides, purple one. The oligonucleotides that don't contain amides have got much less activity. They're down by about 5-fold compared to the amide. This graph on D shows that the toxicity of the different oligonucleotides is quite similar. And the toxicity of the oligonucleotide with LNA amide is not significant because the cell growth is similar to the untreated cells. Some of the phosphorothioate containing oligonucleotides without the amide seem to have at very high concentration more toxicity. So it seems as if this oligonucleotide that's got fewer negative charges is a bit less toxic, but gets into cells far better. Now, this is a preliminary result because it's only one cell line and it's only exon skipping, but it's an encouraging result in terms of what we're thinking about is as follows. If you have an oligonucleotide in which you reduce the negative charge by introducing neutral linkages, does that oligonucleotide work better, i.e. does it get into the cells better? The answer to the question in terms of amide seems to be probably it does. We've repeated this with triazoles, and those results are also encouraging, but there's a lot more work to do in this area to see if this is going to be of any generality, either in different modalities of therapeutic oligonucleotides or different cell types. But it's an interesting starting point. So I want to stop there, but I just want to mention that for people who are interested in oligonucleotide therapeutics, then we've made a webinar on that subject, which is available, and also a webinar on traditional and emerging methods of oligonucleotide synthesis. There's also a webinar that we've made on that subject. And these are available at this URL. And there's also an online nucleic acids book, which contains chapters on all aspects of oligonucleotides for people who are interested in these things. And just finally, I would like to thank members of my group who contributed to this work. There's quite a long list of people that have been working in this area over the last few years. Also in Southampton University, ATD Bio have contributed to the work as well. And I'd like to thank BBSRC and the MRC and the EU for funding. Thanks very much. Thank you.